The goal of HubMap is to create a reference atlas of the healthy human body at single-cell resolution. To achieve that goal, researchers must be able to name and map the trillions of cells and the many distinct cell types that make up the human body. Different cell types perform different functions. While some cell types appear throughout the body, some only exist in certain organs. Authoring and organizing information about all those different cells is the role of HubMap's Anatomical Structures and Cell Types plus Biomarkers, or ASCT plus B, tables effort.
So the ASCT plus B tables are basically a scientific organizational framework where we take anatomical structure, AS, CT, cell types, and biomarkers, and organize this information into an ontologically relevant framework where many different researchers and many different data managers from different backgrounds can collectively see, align, understand, and then use information about biological taxonomies across the whole body.
Take, for example, the renal corpuscle. It is part of the kidney and has different substructures, such as the glomerulus, that are made of different cells. This is the ASCT plus B table for the kidney. Let's zoom in to the renal corpuscle, which includes the glomerular tuft and other substructures. The table also indicates the cell types that make up each anatomical structure. Plus, the table lists the biomarkers that can be used to identify cell types. We see that the glomerular tuft contains 2 cell types, which are commonly identified by specific genetic biomarkers. The ASCT tables are flattened to make them machine-readable so they can be visualized for exploration.
ASCT plus B tables are complex. In fact, each table represents a pathognomy tree and 2 bimodal networks. To help experts answer these tables and reviewers review them, we implemented the ASCT plus B reporter visualization. Using that visualization, you can interactively explore not only the anatomical structures hierarchy, but you can also select, for instance, an anatomical structure and see what cell types are associated with that structure. You can also select a cell type to see what biomarkers are typically used to identify that cell type. In addition, you can use the ASCT plus B reporter to see which anatomical structures have that particular cell type. You can do this not just for one organ, but also for now 10 organs and soon more. For example, you can search for a specific cell type across all 10 organs and start to understand in what organs that particular cell type exists and which biomarkers are commonly used to identify that cell type.
Tables are authored by a team of subject matter experts. Anatomists, pathologists, and surgeons decide on major anatomical structures and their part-of relationships. Single-cell experts work towards standard definitions and characterizations of cell types and their biomarkers. All work closely together to add AS-CT and CT-B linkages based on the rich scientific data that is becoming available. Draft tables are then sent to a second set of experts who review them for scientific rigor, coverage, and quality. Throughout this process, care is taken to ensure that all ASCT plus B tables are in close alignment with existing ontologies like Uberon or CL to support effective collaboration and data sharing. Discrepancies with existing ontologies, for instance, the addition of new terms or term definitions, are resolved in close collaboration with ontology curators.
So one of the reasons why these ASCT plus B tables are important for biomedical research is because there really aren't enough standards in the field for biomedical researchers to use and align and share information on. So this kind of table provides sort of like a Rosetta Stone for people to share what's known about their individual fields using collective ontologies and languages that then can be refined or updated in a sort of common framework for the rest of the field.
Well, I mentioned before how standardization is essential to understanding the information that's being generated and collected by the research community. This standardization makes possible the development of reference spatial systems that provide a means for organizing community data and information by spatially indexing this knowledge with respect to a coordinate system defined over the human body part of interest. These anatomical coordinate systems, or what are sometimes called common coordinate frameworks, serve as atlases for the human body and allow us to pinpoint the location of, for example, where tissue samples are taken, where in vivo biomarker measurements were observed, and so on.
ASCT plus B tables are used to design HubMap's Common Coordinate Framework, or CCF. Specifically, they're used to develop anatomical structure partonomies and cell type typologies in support of semantic search. The tables are also used to design semantically annotated 3D reference organs in support of spatial registration, mapping, and exploration. As new cell types and biomarkers are discovered, the tables will need to be expanded.
The ASCT plus B tables are essential in defining our GPS systems for the human body because they provide the unified place name for every location or position in the body.
Currently, ASCT plus B tables exist for the brain, colon, heart, kidney, and spleen. Tables for other organs are still under development. If you are interested in contributing new tables or reviewing existing ones, please fill out this online registration form, and we will send you information and meeting invites. Or simply contact us at infoccf@indiana.edu.
Hi, I'm Ellen Cordokas, and I'm a biologist working with Dr. Katty Borner's mapping component here at Indiana University for the National Institutes of Health Human Biomolecular Atlas Project, or HubMap. You might wonder how an anatomical structures, cell types, and biomarkers table is constructed. To construct an ASCTB table, we developed a template input form using Google Sheets. The header names are standardized to allow new columns to be added to accommodate varying numbers of hierarchical anatomical structures. Filling in those templates is an iterative process. First, anatomical structures, the cell types found within those structures, and the unique biomarkers that identify those cell types are provided. Next, ontology labels and unique IDs, known as Internationalized Resource Identifiers, or IRIs, are added. Uberon and the foundational model of anatomy are widely used to provide this type of information for anatomical terms. The cell ontology is used for cell types, and the Human Gene Ontology Nomenclature Committee, or HGNC ontology, is used for protein and gene expression derived biomarkers. Standard operating procedure can be found on our Common Coordinate Framework portal. In the future, HubMap will also have spatially resolved lipid and metabolite data that may provide exciting new cell type signatures that can be added to these tables.
The ASCT plus B reporter was developed to support the review and optimization of anatomical structures, cell types, and biomarkers. The tables are authored by domain experts in support of developing an atlas of the healthy human body. The reporter homepage lists major features, contributors, and affiliations. It also provides links to the data tables, the code on GitHub, and the documentation. To go to the visualization, click on the button Go to Visualization. This takes you to the global visualization, where you have the organs in red, the cell types in blue, and the biomarkers in green. This is the organ sheet selector. Currently, there are 10 organs and one additional all organs option, which provides a global view of all the organs. This list lets you select the type of visualization. Currently, there are two visualizations, the tree that you see here and the indented list. This is the refresh button. This button allows you to refresh the data from the flattened Google Sheets in real time. Any change will be reflected from the Google Sheets on the visualizations by clicking this button. Using this button, you can export the tree visualization into different image and JSON formats. PNG, SVG are supported for images. You can also export it as a Vega spec, which is in a JSON format. This button brings in the report. The report computes various statistics of anatomical structures, cell types, and biomarker sets. These statistics can be clicked on to check for additional details. Reports can also be downloaded using the button below. Additionally, if you find any issues, you can report them using this button. This button opens up the debug log. The debug log lists warnings and errors that occur during data parsing and visualization creation, for individual organs as well as for all organs. There are two tabs. The first one displays the logs of the currently selected organ sheet. The second tab displays logs of the current session. Certain messages can be clicked and hovered over to collapse or expand them. Lastly, this button can be used to navigate to the flattened data tables and the GitHub repository for this reporter. On selecting an organ sheet, we can see the titles on the top and the legend. The nodes in the visualization can be hovered over to highlight the links. The nodes can also be clicked for the highlights to persist. Please note that only the nodes in the bimodal network can be hovered or clicked. Clicking on any of these titles will reveal the graph functions. The cell type nodes and the biomarker nodes can be sorted and sized in various ways. They can be sorted alphabetically, which is by default, and by size. The size depends on their degree. The degree is the number of in links and the number of out links. You also can size it by the in degree as well as the out degree. Biomarkers can be sorted alphabetically as well as by their degree. Thank you so much for watching. Feel free to try out the tool for yourself.
It takes a village to construct an atlas. HubMap is collaborating closely with many consortia to construct so-called ASCT plus B tables that characterize major anatomical structures. They also describe what cell types are commonly found in these anatomical structures and what biomarkers are used to characterize those cell types. So you have experts across consortia, across different organs, agree on names for AS, anatomical structures, CT, cell types, and Bs, biomarkers, which could be genes, proteins, lipids, metabolites. And then having those tables, we are also interested to describe the 3D space that these elements occupy. And so we have constructed, in collaboration with illustrators and also with NIH experts, a so-called 3D reference library, where we now have more than 50 organs represented in 3D. And for those organs, we have major anatomical structures that then serve as landmarks for the registration of tissue blocks. Here you have the spleen of the visible human project male. And if you now have a tissue block that you would like to register because you just took it from another spleen, you would move it to the position where it is inside of the spleen. In this moment, you collide with different anatomical structures. These anatomical structures are then associated to the tissue block, meaning you have annotations for this tissue block. And then via the ASCT plus B tables, you also know what cell type populations are commonly found in there and how many of each type. And you also know what biomarkers are commonly used to characterize those cell types. So as soon as you register a block, you not only have annotations for this digital data block, but you also have guidance of what kind of biomarkers you might like to use. For instance, what validated antibodies you can use, or what kind of azimuths or other reference you can use to annotate, for instance, single-cell RNA-seq data. So the next step is to connect this evolving human reference atlas, the ASCT plus B tables and associated reference organs, to the rich data that HubMap has produced, but also data from other portals, but also to recent publications on atlases for different organs. Once we have constructed the atlas, we can understand how cell type populations change as we age. We can see how they change as we go from male to female or across ethnicities, but we can also see how they change when disease strikes, because we see the differences in cell type populations per anatomical structure, per location inside of the human body for these different conditions.
In this video, I'll be showing you how we use collision detection and semantic search within the HubMap CCF registration user interface and exploration user interface. So in the registration user interface, whenever we are registering tissue blocks, we detect what anatomical structures your tissue collides with in the 3D space. And as you move your tissue through the organ, you can see over on the bottom right the anatomical structure tags of all the bounding box collisions that have happened within this reference organ. You can then go through and review and remove things which you know are not in your tissue and potentially add new ones, or to say specifically that these are definitely in there, and they show up as purple. Then when you register, and when you register within the ingest portal in HubMap, this is made automatically available in the exploration user interface, and you will then be able to select an anatomical structure, and it will find all tissues which have that anatomical structure, and that you can explore in the exploration user interface.
Hi there, welcome to the ASCT plus B Reporter. My name is Rishikesh Paul and I'm the lead developer of the ASCT plus B Reporter, and I'm going to walk you through the application. The ASCT plus B Reporter was developed to support the review and optimization of anatomical structures, cell types and biomarkers. The tables are authored by domain experts in support of developing an atlas for the healthy human body. The Reporter homepage lists affiliations, contributors and provides link to the code on the GitHub, the documentation and the visualization. To go to the visualization, click on the button Go to Visualization and then select the organ that you'd like to view. Let's select the heart. On this screen, you have a bunch of functions and the visualization. The visualization shows the relationships between anatomical structures, their cell types and their biomarkers. The anatomical structures and their substructures are colored in red, the cell types are colored in blue and the biomarkers are colored in green. Let's go over the functions in the toolbar. This button lets you toggle the sidebar that you see on the left. Clicking on this will toggle the sidebar. This is the search bar. Click on this and start typing to find structures, cell types or biomarkers. On selecting the entity, a blue rectangle will appear around the name of the entity and it'll be rendered on the visualization. This is the organ selector. There are currently 12 organs that are supported by the ASCT plus B Reporter, along with an all-organ visualization that combines all the other organ visualizations. Clicking on these will take you to their respective visualization. This icon takes you to the data tables from which the reporter gets its data. This icon will refresh the visualization by refetching the data. This icon enables the playground feature. I will go over this later on in the video. This icon will open up the indented list. The indented list is a hierarchical view of the data. This icon will open up the compare feature. Through this, you can link your own sheet from Google Sheets and compare it with the main visualization. I will also go over this later on in the video. This icon will open up the report. The report computes various statistics of the data like you see here. This report can be exported into an Excel format by clicking on this button. This icon opens up the debug log. This log lists warnings and errors that occur during the data passing and the visualization creation, for all organs and for individual organs. There are two tabs. The first one displays the logs of the currently selected organ sheet. The second one displays logs of the current session. This icon will show the export options. The visualization can be exported into two image formats, namely PNG and SVG. The visualization configuration can also be exported into a JSON format. Lastly, this icon will open up options to go to the documentation, the code on GitHub, the terms and conditions, and the policy. Let's now take a closer look at the visualization. The anatomical structures are color-coded in red, the cell types in blue, and the biomarkers in green. The ontology IDs are present below the name of every node. The visualization is interactive, that is, the elements on the visualization can be clicked on or hovered upon to perform certain actions. Hovering on a node reveals a tooltip that shows more information about the node, namely the out degree, which is the number of links that are going out from the node. The in degree is the number of links that are coming into the node, and the degree, which is the sum of the in degree and the out degree. It also shows the ontology ID of that particular node as well as the RDFS label. You can also see that on hovering over a node, the links between that node and its related nodes are highlighted. To persist these highlights, click on the node. The nodes that are not a part of the visualization and the relationship are put to the background to make the visualization cleaner. To deselect, either click on the node or click anywhere in the visualization. To get more information about the node, click on its name. A sheet will pop up from below that will have a small description about the node, the ontology ID, and the IRI link. Let's now look at some of the visualization functions that can be performed. On the left sidebar, there are cell type and biomarker panels. Cell type and biomarker nodes can be sorted and sized. They can be sorted alphabetically or by their degree. They can be sized by their degree, in degree, and out degree. Additionally, biomarker nodes can be filtered by its type. Currently, gene and protein are the two filter types that are supported by the reporter. The reporter also gives you control over the visualization to tweak its parameters. This can be done via the control panel on the left sidebar. This button toggles the visibility of the ontology IDs on the visualization. This slider can be used to change the width of the tree. This slider can be used to change the height of the tree. This slider adjusts the horizontal distance between each bimodal node. And this one adjusts the distance, the vertical distance between the bimodal nodes. Let's take a look at the compare feature. This feature allows you to compare your data to that of the data tables. Click on the compare icon on the toolbar to activate this feature. The form here allows you to enter some data that can be used to compare. You have the table and the description that are optional, a link to your data on Google Sheets, which is mandatory, and then a color of your choice. Here I have a sample data that I would like to compare this to the data tables. So go here, click, copy the link on your browser, head over to the reporter and add it to your sheet link. I'm going to select a color, and you can add multiple sheets, but I'm going to just go with one. And then click on the compare button. So this will get the data from Google Sheets and then create it on the visualization. You will see that all the nodes that are common between your data and the main data tables have been colored in the color that you chose, and all the new nodes, for example this, are added to the visualization with a stroked border around it. This is temporary, and once you refresh the visualization, the data will go away. One thing to note is that in order for the compare to be successful, you need to have the data in the format specified by the ASCD plus B tables, that is this, and also it needs to have a public view, that is anyone on the internet can view this table. Let's take a look at the playground feature. The playground feature allows you to edit and fiddle around with a small version of the visualization, as well as upload your own data and visualize it. Head over to the playground icon on the toolbar and click it to activate the playground feature. Here you have a smaller version of the visualization that is clearer to look at, and you can understand what is happening and how it's working. You have three tabs on the top here. You have the visualization tab that has the visualization. You have the table tab that has a tabular representation of the visualization, and then you have the upload tab. Let's look at the table tab. Here you can edit the data in real time to see how it responds on the visualization. So let's edit this and go back to the visualization. You will see that the name of the node has changed over here. So this is how you can edit the visualization in real time and see how it works. Let's look at the upload tab. Here you can link your own data or any data that is in the ASCD plus B table format and visualize it. Let's go and pick up a visualization data. Here I have the data table for the heart, and once I come back to the reporter and just paste the link and then click on link, it will get you the visualization. Of course, you can change the width and the height, and all of the controls apply to this visualization as well. Remember that this visualization can only be created if the data is public, and also this is temporary, so once you refresh the page, the visualization will go back to the initial one, and the reporter doesn't retain any of your data. So this is solely for visualization purposes. Lastly, let's take a look at the ASCD plus B reporter documentation. So click on the Docs menu on the toolbar on the home page. And this will take you to the documentation of the reporter, where you have all the information about the reporter, how it works, how the different features work, and how you can be controlling those features. I hope this video was helpful. If you have any questions, please reach out to us. Thank you for watching.
We figure that innovation happens when someone runs up against a problem or spots a need. So what problem or need was Vitesse designed to solve or address? Can you tell me just a little bit about how it got started?
Sure. So the major problem addressed by Vitesse is the integrative visualization of multimodal single-cell data sets, and these often contain multiple types of measurements, including spatial and imaging data, quantitative genomic profiles, and gene expression data. And a key feature of HubMap is the diversity of experimental modalities used to measure organ and tissue samples. And we can see this in the long list of data types in the HubMap portal. Previously, visualization tools have focused on visualization of individual data modalities, for instance, with dedicated genome browsers or image viewers. And with Vitesse, we aim to support visualization of all the HubMap data types directly in the data portal.
Excellent. Thank you so much. Could you talk to me specifically about your role in its creation? What role do you fulfill there?
I began working on Vitesse as a summer intern and focused initially on a small component to manage the selection of cells. And later I applied to the bioinformatics graduate program, and now as a student, I've been working on the overall architecture of the tool along with other students and software developers in the Gellenborg lab.
Thank you. Can you talk to me a little bit about some of the challenges that the team encountered in its design or its creation or implementation?
A challenge in developing the tests has been making the tool scalable without compromising on the developer experience or on its serverless architecture. To overcome this, we use the WebGL technology in the web browser to render large data sets. And to load large images and gene expression matrices, we use the ZAR array storage format, which provides compression and concurrent access features without the need for any specialized server infrastructure.
I know you've already kind of hinted at this, but it'd be great to have a statement on this. So what is the most valuable contribution you see Vitess making to its field? You already kind of said this with what problem or issue does it address, but could you just, but what are you most proud of? What do you think is its defining contribution?
We believe that Vitess will be important for publishing interactive visualizations of single cell data as a component that's easy to embed into other websites. With data sets stored in the cloud. And we've demonstrated this use case with the HubMap portal, and we hope that our documentation and tutorials will help other researchers to set up the tests and share their data sets on the web as well.
Great. Yeah, starting with the VH MOOC, which this will be part of, and a lot of people sign up for. So now could you take us through the highlights? Could you give us a tour, an insider's view of this tool?
So this page of the HubMap portal shows the details for a large intestine tissue sample assayed by co-detection by indexing, or the codex strategy, which allows up to 50 protein targets to be visualized within a single tissue section. And so the Vitess component is embedded on this dataset detail page, which allows us to explore not only multiple image regions in this sample, but also cell segmentations, which have been derived by the HubMap automated data processing pipelines. So we can select a different region, and with the image channel controller tool on this left-hand side, we can select a different fluorescence channel for visualization. As we change the cell segmentations, we can see that in yellow, we have a number of cells that the pipeline has segmented for us. And so as we scroll down past Vitess, we can also view provenance details for this data set, and we can also download the raw or processed files. So on this page, we see the main documentation for the Vitess tool. So for researchers who would like to embed Vitess in their own website or use it in the Python or R programming languages, they can come here to learn more. We have a page with documentation and some tutorials. And we also have some demos, including some which are derived from HubMap datasets. For instance, this page, which showcases an attack-seq dataset. So here, we can see the power of Vitess for not only imaging data, but also these other modalities, such as chromatin accessibility. And on this scatter plot, each point is a cell. And we have the cells colored by clusters, just derived by an unsupervised clustering method. And then above this scatter plot and cluster information, we see a genomic profile for each cluster. So these colors correspond to the clusters, and we can zoom in for a more fine-grained view along the genome.
What are we seeing there with the different levels of things there?
So these bars
Yes.
represent chromatin accessibility for each region of the genome. So, and then these are associated with each cluster. So these are aggregate accessibility profiles for each cluster.
And does this allow you to change the view at all, or change the color scheme? And
so if we select our own set of cells, we can annotate this with our own name, and we can also change the color. And in the future, we plan to implement features to view even beyond this aggregate cluster level information, and move toward visualizing a genomic profile for every cell.
Wow, that sounds really great. Very exciting stuff. Mark, thank you so much. I think that's all. That's a lot we can use. And I really, really appreciate you taking the time out of your day. I'm sure you guys are busy, but I really appreciate you stopping by and chatting. And I know that our students will really benefit from this. So thank you very much.
Thank you for interviewing me.
Hello and welcome everybody to this demonstration of the registration user interface, or RUI 1.7.0. The RUI was developed to allow its users to register tissue blocks within reference organs. As you can see, it is a web deployed tool. You can get to the tool yourself by simply going to this URL. So as we get to the website, we are being greeted by this modal window where we're being asked for some basic information. So first we have to type in our first and last names. Of course, the browser remembers what I previously entered, so that is a fairly quick step. Then I have to choose the donor sex for my first registration. In this case, I'm going to go with female. And then we have to select an organ. We are now supporting 11 organs in the RUI. You can see all of those listed here, so you can press these little arrows to get through the carousel here. And for this case, let's just go ahead and use the lungs. We click the lung. And then you can see that we can start a registration. This button then becomes blue. So I click, and then the scene is being set up for me. So the RUI consists of 3 panes. Here on the left side is the anatomical structure pane. In the middle is the 3D pane, and on the right side is the manipulation pane. So on the left side, we have, again, the donor sex, where we can switch any time if we have to. For now, I want to stay with the female version. And then we have here a list of anatomical structures. And so this allows me to set the opacity for structures that you can see here in the 3D scene. So for example, if I wanted to see more of the trachea, I can increase the opacity of the trachea here. We can see the trachea here. And then as I move this opacity slider around, we can see that I can change the opacity of this anatomical structure. I can also turn it off completely by clicking this eye icon here, and then it just disappears. Then similarly, I can do the same thing, for example, for the bronchus. So I can set the opacity all the way up, or I can remove it entirely. Another thing you can do is you can reset everything to the default values by clicking the circular arrow here, and then everything returns to the default, which is a 20% opacity for all anatomical structures. The next thing I would like to demonstrate are the common extraction sites in the previously registered blocks. Currently, I have neither for the lung, so I'm going to switch over to another tab real quick, where I'm going to show you the same thing with the large intestine. So here we have the large intestine, the female version in fact, and here on the left side you can see that we have a set of common extraction sites. These are visual landmarks where we identify together with experts that these are frequently used extraction sites for tissue. So in this case, we have 4 common extraction sites. We have the ascending, descending, sigmoid, and transverse colon. And here, just like with the anatomical structures, I can hover over this icon here, and then I can make them visible. So I click this eye icon here, and then I do the same thing down here. I do this for the sigmoid colon as well, and then for the transverse colon. And then you can see these green markers appear. And these green markers are the common extraction sites. Then of course I can also adjust the opacity again. So that in this case now I would have full opacity for all 4 of these. So again, these common extraction sites are markers for commonly used extraction locations. Now to demonstrate another piece of functionality, I would like to show you that you can view previously registered blocks. So we have this button down here, previously registered blocks. If you click this button, then we are using information from your browser to display previously registered blocks for a particular organ. So for example, you can see here that I have done quite a few previous registrations on this particular machine for the left male kidney. So now let's go back to the lung. So this is the anatomy pane on the left that really allows you to customize what you see here in the 3D pane. So the 3D pane is where the actual 3D interaction happens. So you can see that we are looking at the organ from a certain angle, from a certain camera perspective. And this perspective is also shown here on the right with this little mannequin symbol. So currently we are in the anterior view. You can see that we are also looking at the anterior. We are having an anterior view of the mannequin here. And now I can change my camera positions. For example, I can go from anterior to right. And then the camera moves to the right side of the reference organ. We can see also that we are now on the right side of the mannequin. You can also go to the left or to the behind. Here we go. Let's go back to the anterior for now. So this is how you control the camera in order to customize the viewpoint that you have with regards to your organ. So that's a registration mode, and then we have also an overview mode. So if I click this little toggle switch here, we are making a switch in the way we control our camera. So previously we could only control our camera with these radio buttons. Now we have a fully functional orbit camera. What does that mean? If you click and drag your left mouse button, you can rotate the camera around. Just try that out, it's kind of fun. So yeah, left mouse button, press and hold. Now, if you click and drag your right mouse button, you can pan the camera, so you can move around the camera. And then finally, if you use your mouse scroll wheel, you can zoom in and out. This might take a little bit of practice, but it is a really neat way of exploring a 3D scene, basically with a lot more freedom that you would have in the registration view. However, in order to actually perform the registration, we have to go back into registration view. So I click the toggle button again, the toggle switch again. And now on the right side here, we can see a little yellow cube that turns blue when I hover over it. This is the tissue block that we want to register. So if I click and drag this tissue block, it follows the mouse, okay? And then let's say we have a piece of tissue that comes here from this upper part of the left side of the lung. I can go ahead and I can click and drag this in here. And then of course I'm gonna have to make some adjustments. So I can now, for example, switch to the left view like this. And then I can move this up a little just to make sure that I am in the place that I want it to be. Okay, and then we can use the 3D preview mode by clicking the toggle switch again to zoom in, rotate around, and ensure that I really have registered what I wanted to register. So one thing, for example, that I might want to change here is the size, and this is where the manipulation pane comes into play. So first here we have the tissue block dimensions in millimeters, so that allows us to simply set the dimensions of this cuboid. Let's go with 8 by... Let's do 6 plus 1 millimeter. There we go. And now, as you can see, it resizes quickly. Then I can input the number of tissue slices. So let's say, for example, here's thickness in micrometers. Let's say the thickness is, say, 10 micrometers, and I have 100 slices. There we go. And then we can also adjust the rotation here. So each of these sliders lets you rotate the block around one of its axes. So in this case, let's rotate it around the x-axis, like so, such that it is as flat as possible towards the upper part, like the upper boundary of the organ here. And then, and this is an interesting one, we have the anatomical structure tags, and this kind of brings us full circle back to the anatomical structures here. So let me go back into registration view. So as I grab the tissue block here, I'm going to move it out for a second. And in a second, I'm going to move it back in again. And I would like you to watch what happens here in this anatomical structure tags area. So I grab the tissue block and I move it in. And then as I get into the lung, we see these tags here appear. And so we are employing a system called collision detection, which is fairly common in 3D world. Imagine that you're watching a Hollywood movie and two vehicles are colliding, okay, like for a really cool action scene. Obviously, it is computer generated, but the computer has to have a way of knowing that a collision just happened. So there is a collision detection algorithm running. And then, of course, in a Hollywood movie, you get like really cool special effects. In our case, you get something very simple. You just get an anatomical structure tag. So this tells us that, hey, our tissue block has just collided with these anatomical structures. And you can see that these anatomical structures correspond to structures that we have here. And you may find that, for example, some of these collisions may have been not correct. We are using the boundary boxes of each of these 3D objects. So the collisions might not always be 100% accurate. And in this case, you can go ahead and you can click this X here, and then you can just remove the anatomical structure tag, and you can also add some. So for example, if you think that only parts of the registration so far are of the anatomical structures are correct, you can just type in one of these structures up here, click the little checkbox and then the plus, and then it appears here, and then you can see that it is marked as added by the user. For now, let's leave it out though. All right, so now let's go back into the 3D preview mode again, just to verify one more time that we did a good job here. Okay, so let's say we wanted to verify that we are actually really inside of the organ. Then I would go ahead to the anatomical structures, go to the lung here, and set the opacity all the way to 100. And then I would make sure that the tissue block is completely enclosed within the outer surface of the organ, which seems to be the case here. Let's go back into registration mode, the anterior view, and enjoy this beautiful lung here. All right. Now we have created this registration data, but what do we actually do with it, or how do we actually get it out of there? So you can click this review and download button down here when you are done, and then you are presented with this registration review. So this contains all of the data that we just generated, or most of it. So we have the first name, last name, we have the name of the reference organ, then we have some geometric information such as the dimensions, position, and rotation of our tissue block. We have the anatomical structure tags, a timestamp, and then a unique alignment ID. So I'm going to go ahead and I'm going to download this registration data to my machine. Here we go. And now let's just have a little look at what this data set actually looks like. So here I now have the JSON file open that I just downloaded. So JSON stands for JavaScript Object Notation. And that is the format that we chose to capture and also share the data with some of the backend tools in the CCF and across HubMap. So you do not really need to understand any of the things that you see here. This is just to show you what the data actually looks like. You can see that all of these things here, these correspond to some of the data that you saw inside the RUI, okay? And yeah, this is essentially what you create. And so you have that then on your machine. Let's go back to the RUI here. So now let's say that you created a registration and it was a tissue block that you extracted many times, and you just want to reuse it multiple times for multiple registrations. So let's go ahead to a blank slate of the RUI. So we are again being presented with the modal. So I'm going to type in my name one more time. And now let's say that I want to just recreate the registration that I just did. You can go to this blue Upload Previous Registration Data button. Then you select your registration data. And then you can see that everything is being reset just like it was before. So we can see our little tissue block just automatically resized and rotated and got put into place here inside the lung here. Finally, the last thing I want to go over is this info button up here. So if you click the info button, you are being taken to a little section about the usage of the RUI with a link to this video, by the way. And then we have an about section down here that explains a little more about the background. We have a link to a standard operating procedure that explains everything that I did here with in written form, and with a little more detail depending on where you're coming from when you're using the RUI. And then we have acknowledgements here on the bottom. Okay, this was a demonstration of the RUI 1.7.0, which is a tool that allows users to register tissue blocks into reference organs. We are very glad that you are going to use that tool, and please reach out to us for any questions. Thank you. Hello everybody, and welcome to this demonstration of the CCF Exploration User Interface, or EUI. The EUI is a web deployed tool, so you can go to the tool yourself simply by typing this URL into your browser. The EUI was designed to visually explore, hence the name, the spatial and semantic context of registered tissue blocks within HubMap, or for outside HubMap as well in certain cases. So the EUI consists of 3 panes. We have a partonomy and ontology pane on the left, we have a 3D pane in the middle, and we have a details pane on the right. We also have some UI tools that help you adjust your workspace. So for example, here we have a full screen button. If you press that, you go into the full screen mode for the detail section here. We have this little button here to switch into dark mode. If you, like me, prefer to work with a dark mode, this is the way to go. I will, however, in this demo use the light mode because that is the default that most people are probably going to use. So what the EUI does is it shows you the reference organs within the male and female bodies and the tissue blocks that have been registered for these organs so far. So let's start with the 3D pane in the middle. I have an orbit camera that I can use to go through the 3D scene. If you click and drag your left mouse button, you can rotate the camera. If you click and drag your right mouse button, you can pan the camera, like move it sideways. And by using the mouse scroll wheel, you can zoom. So let's go ahead, go over to the male body here and just zoom in a little bit. And then as we go closer, we can see existing tissue blocks. Those are actual tissue blocks that have been registered by users into our common coordinate framework. We can see that there's quite a few here across these organs that we see. Here in this carousel on the top, you can see all the 11 organs that we currently support. You can also see those little numbers below each organ that show you how many tissue blocks we currently have. And furthermore, we can also see that some of them are active and some of them are inactive. So for example, the brain is currently not visible, neither is the large intestine. However, the large intestine has 4 registrations, so let's click on it, and then the colon appears in both reference bodies. So let's go again to the male here. Here we go. And now here we can really nicely see those pieces of colon that have been registered for the male colon here. I'm going to turn this off again. I'm going to zoom out to give us a nice view of the 2 reference bodies with the organs inside. So now let's look at the partonomy and ontology pane here on the left. Here I can search for ontology terms. So for example, let's say I am specifically interested in the ascending colon. I can type this in and then I can see that there's an autocomplete function here. So I go to ascending colon and then the partonomy here opens automatically to the ascending colon. We can see that we have 1 tissue block for the ascending colon, which we saw just a second ago. All right, so let's collapse this again. This shows you just how many tissue blocks are available for each of these organs and then for each of these anatomical structures accordingly. Another thing that we have here are filters. So I can click this filter symbol up here, and then I can select a whole bunch of parameters or adjust a whole bunch of parameters to narrow down my search. So for example, let's say I am only interested in registrations for the female body. Let's say age 18 to the age of, let's say, 50. And then I can click this apply filters button. And then we can see that we only see the female body because that's what we defined as the sex. And then we can also see that the details pane here on the right got smaller, because those are the ones that we have for from female bodies age 18 to 50. So now I can zoom in a little bit. Let's focus here on the organs. And then here we see the actual registrations. Okay. So I'm going to go ahead and I'm going to click this checkbox here for the first registration and this one for the second. And then we see here that these have been highlighted with orange and blue colors respectively. We can see one of these registrations. The orange one is for the spleen and the blue one is for the kidney. And then if we click on these buttons here, then we see a lot more information about the actual registration that we have here. And then if we click on these thumbnails, we can see the actual assay that was performed. So we can see here 10x. So let's click on this. And then the actual data set opens. Okay, we can also see the identifier of the data set and a whole bunch more information. And then we can click this open a portal button, and then a new tab opens and we can look at the data inside the HubMap portal. And I can click this X here to leave this detail view again. Let's zoom out again. Let's collapse these. And then I can also select more, and we can see how all of these colors are being applied programmatically, just so we have a nicer way of distinguishing the tissue blocks from each other. Another thing I would like to demonstrate is, again, the enhanced details pane. So as I expand this area here, we see some metadata information. So we see that it was registered on June 9th, 2020. We see the the operator. We see the TMC where it came from, the dimensions, etc., etc. And then here we have this blue line, which stands for the Z-axis. And this is the axis along which individual tissue slices have been cut. And then you can see here these little vertical lines here that symbolize the tissue sections that have been taken. And then, of course, if a tissue block has more than one assay or modality performed on it, you can see more thumbnails. Just like you can here, for example. This is a whole bunch of modalities that exist for this particular tissue block, which is here in the right kidney towards the upper part. Of course, if I click on this, I can highlight this specifically, and I can do that for the other ones as well. All right, this was an introduction to the Exploration user interface. We looked at the parthonomy and ontology, we looked at the 3D scene, and we looked at the details pane. And I hope that you find it straightforward and easy to use this Exploration user interface. If you have any questions, please feel free to contact us anytime. Thank you so much.